workshop called Digital Image Basics. Before we begin working extensively with multiplex immunofluorescence images in QuPath, it's important to understand what a digital image actually is and how it is generated. Many image analysis decisions depend on understanding how image data are acquired, stored, and displayed. In this module, we'll cover the fundamentals of image formation, pixel values, data types, image visualizations, and histograms. These concepts will help you interpret fluorescence images more accurately and avoid common mistakes when analyzing image intensity data. In this module, we'll address three key questions. First, how are fluorescence images captured? Second, what exactly is a digital image? And third, what information can we readily interpret from image data? By the end of this session, you should be able to identify the major components involved in creating a digital image, describe images in terms of pixels and dimensions, and understand what lookup tables or LUTs do, and recognize several ways to summarize image information. Let's start with a simplified view of a microscope. There are three major components involved in image formation. First is the sample, which is the biological specimen we're interested in observing. Second is the objective lens. The lens collects light emitted from the sample and focuses it towards the detection system. Third is the detector, such as a charge-coupled device or CCD camera. The detector converts incoming light into electronic signals that can ultimately be stored as a digital image. Although modern microscopes are much more sophisticated, this simplified model captures the essential principles behind digital image acquisition. Now let's zoom into the detector itself. When photons strike the camera sensor, some are converted into electrons. These electrons are then measured and converted into numerical pixel values. A critical point is that not every photon generates an electron. This conversion process is imperfect. Because of this, measured fluorescence intensity may not perfectly reflect the true number of emitted photons. This is one reason why quantifying fluorescence signals can be challenging, and why experimental conditions need to be carefully controlled. Several factors influence the pixel values we measure. One factor is exposure time, or how long the detector collects photons for. Longer exposure times capture more photons, resulting in higher pixel values. Another factor is the numerical aperture. Here, the numerical aperture, or NA, of the objective lens, if it has a higher NA, collects light over a larger angle and allowing more photons to reach the detector. A third factor is detector sensitivity, often described as quantum efficiency. More sensitive detectors convert a greater proportion of incoming photons into measurable electrons. These factors can significantly affect image intensity even when the underlying biology remains unchanged. This leads to an important caution. Individual pixel values are rarely meaningful on their own. Instead of focusing on a single pixel, we usually examine groups of pixels, regions of interest, cells, or tissue compartments. In image analysis, we are often interested in relative differences between samples rather than absolute intensity values from isolated pixels. So, always interpret pixel values within their biological and experimental context. Digital images can be thought of as arrays of numbers. Each pixel occupies a position within a grid and stores a numerical value representing the detected signal intensity. In fluorescence microscopy, these values correspond to the amount of light detected at each location in the image. Thinking of images as arrays is especially useful because image analysis software like QuPath primarily works with these underlying numerical values, rather than the visual representation we see on the screen. Images can be described in different but equivalent ways. For example, an image may have two dimensions where each dimension contains 20 pixels. We could describe this image as having a shape of 20 by 20 or simply say it contains 20 by 20 pixels. Understanding image dimension becomes increasingly important when working with large whole slide images or multi-dimensional fluorescence datasets containing additional channels, z-planes, or time points. Another important note is that some coding programs start their counting from 0 instead of from 1. So knowing which of these is important, and hence you can describe an image by its dimension from 0 first, or from the first dimension being from 1. Next, let's discuss data types. Data types determine how pixel values are stored and therefore what range of values is possible. Three important concepts are dynamic range, intensity clipping, and intensity rounding. Dynamic range refers to the range of values that can be represented. So here on the left we can see a diagram of some different bit depths that a digital image can store a pixel value in. A bit depth refers to a binary number and the number of zeros or ones that form that binary number. Each of those numbers are coefficients to 2 to the power of the relevant index. For example, in the top we have 0 represented by all zeros, meaning that every coefficient in front of a 2 to the power is 0 and so sums to 0. Whereas if we have 1 in the first number, then this refers to 1 in front of the 0th power of 2, and 2 to the power of 0 is then 1. And so we can go through changing these zeros and ones in the binary number to form different numbers between 0 and, in this case, for an 8-bit image, up to the value of 255. Now, intensity rounding happens when values are converted between formats and precision is lost. For example, the maximum value for a 16-bit image is 65,535. Yet there are more numbers between 0 and this, and so when maybe an image is converted from a 16-bit to an 8-bit, some of those numbers are merged into other integer values. The other situation that we need to consider is intensity clipping, or sometimes referred to as saturation. This occurs when values exceed the maximum representable limit and are therefore truncated. Therefore, data is lost because we don't truly know if the maximum value is the maximum value, or there were higher values that the fluorescent signal would have been. These concepts affect both image quality and quantitative analysis, and should always be considered during image processing. Now let's discuss lookup tables, or LUTs. A LUT determines how numerical pixel values are displayed visually. The key point is that LUTs change appearance, not the underlying data. For example, an image may be displayed using grayscale, a pseudo color palette, or a fluorescent style color map, while the pixel values remain exactly the same. Understanding this distinction helps prevent confusion when comparing images that appear different on screen. Brightness and contrast adjustments provide a common example of LUT manipulation. When we change brightness or contrast, we alter how pixel values are mapped to displayed intensities. Importantly, these adjustments usually affect only the visualization. Unless the image is explicitly processed and saved with modified pixel values, the underlying data remain unchanged. This distinction is critical for maintaining scientific accuracy during image analysis. For this exercise, we're going to open the fluorescent cells sample image we had from a previous module. We're going to navigate it in the viewer and then go to the View and Brightness Contrast tool, which will open a window and where we can then manipulate the LUT using the various options available within that brightness and contrast window. Take some time to explore the available controls, including channel selection, saving display settings, grayscale display options, background inversion, logarithmic histograms, channel minimum, maximum values, view gamma, auto adjustment, and reset. As you experiment, notice how dramatically the image appearance can change, even though the underlying pixel values stayed the same. I'll pause here to give you a chance to try this yourself. I'll now demonstrate how to perform these steps of the exercise. We're in the QuPath software and you can see that I have the fluorescent cells image open in my project. We'll now go to View in the menu bar and to the Brightness Contrast tool. We'll see that a new window has appeared and we can move the window somewhere else in the image or make it larger as well. We can see that we have the different channels, and often when we open other fluorescence images, the channels will refer to the markers that were used in imaging. This is a consideration about the metadata. But in our case, we simply have the red, green, and blue channels. We can toggle all channels on and off using the show toggle here. And we can also toggle individual channels on and off too. If we want to look at a channel in grayscale, then we can select this button here, Show Grayscale. And now we can look at each of the images using a comparative lookup table. But we can only do this for one of the channels of our image, because we can't distinguish between which channel is contributing. We can also save the settings for our brightness and contrast by clicking Save, and here we'll be prompted to give a name to our display settings. So in this case, we'll simply call it green only. Click OK. And now in this drop down, we can see that green only is a display setting which we can toggle on. For example, if we have all of our channels on again, we can simply say green only and we'll revert to those display settings. As well as showing the lookup table in the intended color, we can also switch the colors around so that white is now displayed for the lowest pixel values, but still keeping green as the highest. But also, if we don't want to have green as the color of this, we can also select the colors as, next to the name of the channel and pick a different color instead. Let's say cyan. You can see that the name of the channel did not change, but the color has. And this again we can save in our display settings. We can see towards the bottom of the brightness and contrast window we have what's called a histogram, and we'll cover this later on in the module. But we can adjust the channel min and maximum for the selected channels. So now that I'm selecting the green channel, we can then change what is then considered all black, leaving only the highest value pixel as in between that all black and the brightest of green. And we can also change the channel max so that the brightest green is shown for lower values of the pixels. The gamma refers to how your computer monitor is being displayed, and you would have seen that as you changed it, a warning comes up saying that this is maybe something you did not intend to do. We can simply hold the shift and click on this warning to reset back to 1. Now, in QuPath, we also have some buttons to automatically display for a particular channel. So here we have auto, which will then move the channel min and max to values it has automatically identified as a nice way to visualize. We can also click reset, which will then revert to the lowest pixel value and highest pixel value of our image. Remember, the highest pixel value may not be the highest possible value of our pixel depth. But if we want to adjust it to a value outside of the minimum or maximum, we can double click on the value next to the channel min or max and reassign it a different value. In this case, our values don't have anything higher than 255, so when I move it to what would be the dynamic range for a 16-bit image, we have pretty much everything black. Now that we've interacted with the lookup table in QuPath, I mentioned about a histogram in the window. When comparing images, checking every pixel individually is possible, but is usually very inefficient. Instead, we often summarize image information using statistical measurements, such as the mean, standard deviation, minimum value, and maximum value. Another useful tool is the histogram. Histogram provides a graphical summary of the distribution of pixel values within an image. Statistics and histograms offer fast ways to evaluate similarities and differences between images. A histogram is a graphical representation of quantitative data. To create a histogram, pixel values are grouped into intervals called bins. We then count how many pixels fall into each bin. The resulting graph helps us visualize the overall distribution of intensities within the image. Histograms are particularly useful for assessing contrast, identifying background signal, and detecting saturation or clipping. However, remember that they summarize value distributions and do not tell us where specific pixels are then located. Here we see that different images can be displayed using the same visualization settings, while producing different histograms. Although the display method remains consistent, the underlying data distributions differ. The histogram therefore provides information about image content that may not be immediately obvious through visual inspection alone. This makes histograms a valuable tool for quality control and image comparison. In contrast, this example shows the same image displayed in different ways. The visual appearance changes because different LUTs or display settings are applied. However, the histogram remains unchanged because the pixel values themselves have not changed. This reinforces a central concept. Display settings alter visualization, whereas histograms reflect the underlying image data. Now, can the same histogram mean that the image is then the same? The answer is no. Two images can have identical histograms while containing very different structures and spatial arrangements. Histograms only describe the distribution of pixel values. They do not preserve information about where those pixels occur within the image. As a result, histograms are useful summaries, but cannot fully capture image content. Let's revisit our learning objectives. We discussed how fluorescent images are captured through interaction of samples, optics, and detectors. We learned how digital images are organized as arrays of pixels. We also explored LUTs, display settings, and statistics in histograms as tools for understanding image data. These concepts provide the foundation for more advanced image analysis workflows in QuPath. To conclude, there are several important take-home messages. Multiple experimental factors influence image intensity, making absolute intensity comparisons challenging. Two-dimensional digital images are composed of pixels organized in a grid. Each pixel stores a discrete numerical value, and the pixel depth determines the maximum possible value and the range of values it can possess. Lookup tables change how images are displayed without changing the underlying data. Finally, histograms provide useful summaries of pixel value distributions, but contain no information about the spatial organization. Understanding these principles will help you make more informed decisions as we move into visualization, quantification, and analysis in QuPath. Thank you, and let's hope you join us for our next module, Colors and Channels.